Okay, guys, let's do this. Exam 1, get up in recitation. That'll give you the opportunity to ask me any questions you have. Today's recitation, we're going to make up this noting material. Don't forget that, so you've got 2 quizzes to deal with today. Hopefully you're prepared and you don't have to really put too much effort into it. In the alternative is you just skip it and that's one that you drop. But, you know. Exam 2 is week of 4-1. This is the distribution of the first exam. That's really awesome. That's really good. I was actually, sometimes I get these two-tail, kind of like these multimodal distributions, and that's what I thought I had, but when I actually looked at it, we've got a lot of really good grades. Some people are really struggling. Those who are, you know, in the 60 and below, make sure you see me. And we talk about your study habits. A lot of people have already seen me. And what we're coming up with by analyzing how questions were answered is that people are memorizing, and then they get confused and jumbled up. Whereas if you focus on understanding and using logic, rather than just stuffing your brain full of stuff, then it'll be easier to answer these questions. But this is awesome. I actually aim for a 75 average, so this is fantastic. Okay, a quick message about fire safety. So as you'll see here, you have candles and a sleeping person. Okay? And let me show you what that caused on my street on Monday night. So this is the house. Let's see. This yellow house is the one that was on fire. And that's my house. And that was 1 in the morning. You only see, I think, 3 fire trucks. There were 4 guys. It was not fun. And so I smell like a barbecue right now. And everything smells really bad because our house filled with smoke. But I want you guys to stop and be thankful for having your home and the people you love and everyone is safe in your life. Because it's when these things happen that you stop and you reevaluate and think about what's important. And I know it's really easy to get caught up on grades, but who the hell cares about grades if you're dead, right? In all honesty, right? So you have to keep things in perspective, and it's really easy to get your anxiety out of control when you're worrying about grades and tests and what you're doing in your future. But if you don't realize that the world is bigger than that, then it's not good. So the reason I say candles and sleeping is it was started because the house that went on fire, it's a bunch of young students, one of which had a candle in her room and she fell asleep. So I know everyone loves those smelly candles, but just really be careful. Because that whole, they're all homeless now. Well, luckily most of them have boyfriends and places to go, but really scary. So that's my story, and somehow I have something to teach you today. I can't, just like you guys tell me about the drama in your lives, I tell you about the drama in my life. Because, we all have to understand what each other's going through. It's a full story. Anyways, back to Cell Cycle. On that note, if anyone wants to come in my office and smell my coat. I walked into my office yesterday and I was like, oh my God. All I did was leave my bag and my coat in there, and my whole office smelled like smoke. And I was like, really? Not even at home. Anyways, let's just hope I don't need to move out of my house, because that's what my neighbors are having to do for it to be cleaned, and that will really suck. So far, we've been studying cell cycle. On that note, so sometimes when things get stressful, it's really nice to have work to dive into. So that's always good. So we talked a little bit about cell cycle regulation. Oh wait, any questions about fire safety? We all know, don't fall asleep with burning candles. Cell cycle regulation, we talked about that. We talked about checkpoints. We've been talking about S phase. We talked about DNA polymerase, structure, function, proofreading ability. We've talked about the different enzymes involved in replication. We talked about initiation in bacteria. And we're going to talk we talked a little bit about it in eukaryotic cells, but we're going to move forward on that. We talked about origins and how they differ in bacteria versus yeast versus eukaryotic cells. Yeast is a little bit different because it has the ARS. we talked about regulation of origin firing, and that's where we ended on Monday, when my house still smelled like a house. Okay, I'm a little traumatized, just a little. Running out of houses in the middle of the night with kids, not fun. Okay, so here's the big picture on initiation. Initiator proteins bind to DNA, and it binds before DNA replication is ready to begin, and then the complex is activated. Big picture, basics. Oh yeah, sorry. Better? Okay, sorry about that, guys. Okay, so this is the big picture. It's easy to get bogged down with DNA A, B, C, blah, 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 blah. Initiator proteins bind to DNA, and then it gets activated somehow. Right? And so I'm bringing this back in because we talked about it before in the context of the CDKs and cell cycle, but I really want you to be kind of meshing it with the true activation of DNA replication. Okay? So we're talking about the mechanism of DNA replication. Don't forget, it's these cyclin CDK complexes that regulate that. Okay, so how does cell cycle regulate DNA replication? This is a very big picture question that may very well, as big picture as you can get when you're talking about the cell cycle, right? It's kind of like, I was talking to someone, I was like, yeah, I try to keep a big picture, and then I realized I really don't, right? Because I'm kind of like stuck in a cell. Big picture is like looking at the world. Anyways, okay, so the origin recognition complex, it binds to the origin. We've seen this, right? And then that all happens in M phase G1. Then CDC6 and CDT1 recruit MCM helicase. So these are helicase loaders recruiting helicase to the origin recognition complex and loading it onto the DNA. Right, so this is called, I think, licensing. So then really what needs to happen is you need to activate this helicase complex in order for DNA replication to begin. So it will become an active helicase during S phase. So load it on, activate it, yes?
So does the ORC contain the same domains on both sides of the complex in order to recruit the same proteins on both sides?
Does the ORC contain the same domains on the same on opposite sides to recruit? Okay, so you're seeing the symmetry, and you're asking if the organization of the ORC is symmetric, right? I'm guessing, probably. I'm not 100% sure. Logical reasoning. So there are two levels of regulation here, right? One is helicase loading, and the other, so that's the pre-RC assembly, so that's this assembly of this whole complex. And the other is helicase activation, and that's what SCDK is triggering. Okay, so it can load, it can sit there, but it will not be active. So regulating pre-RC assembly is the reason that S phase only happens once per cycle. So we talked a little bit about this in bacteria the other day when we talked about origins being methylated and hemimethylated, and you need full methylation to initiate DNA replication in bacteria. So that's how bacteria regulate S phase. And this is how eukaryotic cells regulate S phase. And there are a few different things that are happening, right? You're going to get inhibition of this complex by geminin. So geminin inhibits helicase recruitment. It's actually degraded by APC at the end of M phase. So that's what allows this complex to form. But then once APC is activated again, then you're going to, once APC is inhibited, when it's shut off, you're going to get accumulation of geminin, which is going to inhibit pre-RC formation. You'll also get You'll also get replication-dependent CDT1 degradation. This is one of these components of the pre-RC, and you'll actually have degradation of this protein, and that's because CDKs are targeting it for degradation through phosphorylation. And so this was the major concept that I had talked about before, CDK-dependent inhibition of pre-RC formation, because CDKs are kinases that are phosphorylating things, and those things are frequently either inhibited or degraded through phosphorylation. So phosphorylation by CDKs of CDC6, CDT1, MCM2, ORC, all these components of the pre-RC, they're phosphorylated, and that results in degradation or inactivation. So inhibition of recruitment of all these proteins to the origin is going to prevent cell cycle DNA replication from progressing. Makes sense, right? If you don't want something to happen, then either degrade or inhibit all the different proteins that need to assemble there, right? Pretty logical. Okay? Questions? This is all kind of a review of what we already learned, but I'm trying to kind of put it together with DNA replication. Okay, so another way to look at it, phosphorylation by CDKs inhibits pre-RC formation. Okay, obviously not by SCDKs, because SCDK is going to activate DNA replication, but remember there's a different CDK for every phase of the cell cycle, right? So ORC, right, it can bind to origin pretty early on. See, this is G2, M, G1, can always assemble there. But recruitment of the other components of the PRC, it's inhibited by CDK-induced phosphorylation and degradation of different components of that complex. CDKs inhibit CDT1 and MCM assembly at the origin by promoting export from the nucleus. So you can get phosphorylation of CDT1 and MCM and actually export these proteins from the nucleus. If they're not in the nucleus, can they assemble at origins? No, right? So kick them out of the space that they need to be in, and they won't work. Yes?
Is the ORC the same as the initiation protein?
Yes, so you're comparing it to bacteria? Yeah, so that would be DNA-A. Similar in function? Yes?
Are there proteases in the nucleus?
Okay, the question I'm getting is, are there proteases in the nucleus? What's your thinking?
My thinking is it kicking out the CDT1 and MCM so it can like save it for later, or is it just sending it out to be degraded?
It's sending it out of the nucleus, so it's not available to bind to the DNA. So it's just relocalizing it, right? By obscuring a nuclear localization signal and showing, displaying a nuclear export signal.
So will it be brought back in?
It will be brought back in when it's needed. And this is phosphorylation dependent. So you can imagine it probably has a nuclear import signal and export signal, and probably the nuclear import signal is being phosphorylated, so it's not recognized. Questions?
So just to clarify, the MCM you're talking about here is the same as the MCM2 side.
Yeah, the helicase. Yeah. The MCM I'm talking about here is helicase. So I pull a lot of these figures from all over the place, and so sometimes the wording is a little different. Yes. Any other questions? Yes.
So what's the difference between ORC and pre-initiation complex?
ORC is the origin recognition complex. That's the first step in formation of the pre-initiation complex. So the first thing is the ORC binds to the origin, recruits all these other proteins, and that's the pre-RC complex when all the other proteins are recruited, and then it's just an issue of triggering activation. Okay, so it's like step one. Okay?
So it's part of the pre-initiation.
It's part of the pre-RC. Yeah, and then the pre-initiation complex is recruited, which is polymerase and all that, and then you have the formation of the replication force. Okay? Any other questions? Who loves this? A few people. Okay, so phosphorylation of ORC by CDK prevents interaction of ORC with CD21. So you can actually get phosphorylation of ORC by a CDK. So you can see the CDK with the little T here, that means inhibition. So if you phosphorylate ORC, it's going to prevent interaction with CD21. And remember, CDC6 and CD21 help to load helicase. So if you're preventing interaction with CD21, you're preventing recruitment of helicase. Helicase isn't loaded. You're preventing pre-RC formation. Okay, so it's a bunch of these steps that need to happen, and it's really about going back to the big picture because it can seem overwhelming with a bunch of proteins on a page here, but it's really about assembling that helicase and then activating it. Okay, you're recruiting helicase, activating it. This is about really recruiting the helicase. Okay, if you don't recruit your helicase because you've either degraded components of the complex required for recruitment or you've sent things out of the nucleus, you definitely can't activate it, right? This is logical. So try not to get overwhelmed with the detail and think about the big picture. Recruitment and activation. Okay? Yes. You give me quizzical eyes and I feel like...
You can cover it, sure.
Okay. Whenever people have confusing looks on their face, I feel the need to... Yes?
For number one, when it says RC can bind origin as early as G2, and then it's in the N space before G2.
G1, S, G2.
So would G RC bind before the N space?
Remember, okay, so you're thinking of it linearly. So remember, cell cycle is a circle, right? So it's not linear, right? So G1, S, G2, M. Okay? So anytime after S, you can get ORC starting to assemble at origins again. So anytime after, ORC might start to bind to an origin. But it's not active. So remember, if it's here at any time here, it's sitting there waiting to recruit helicase, and then you get activation of helicase. And then you get S phase. Okay? Did that answer your question? Okay, any other questions? Good. Okay, so phosphorylation by S-CDKase activates the pre-RC and establishes replication forks. So we haven't even established replication forks at this point, right? We just have this all this stuff sitting at origins just waiting. And S-CDK, remember S-CDK gets turned on because of what? S-cyclins, right? So S-cyclins were building during all this time. They get to a point where they then can interact with S-CDKase, activate S-CDKase, then they can phosphorylate things and turn them on, or turn them off depending on what needs to be done. But phosphorylation by S-CDKase activates this pre-RC and establishes replication forks. So that's what you're seeing here. This pre-RC, this is your ORC and your helicase and your CD21 and your CDC6. All this stuff is just hanging out. As CDK comes along, starts phosphorylating things, and then disassembles parts of this, leaving your helicase, and then you get your recruitment of your pre-initiation complex, and then polymerase, primase, clamp loader, all those things that you need for DNA replication to happen. And then you have activation of DNA replication. Okay? Questions about this? So phosphorylation of CDC6, that allows it to be recognized by SCF. Remember we talked about that ubiquitin ligase that has the F box, and it recognizes phosphorylated substrates. So phosphorylation allows CDC6 to be recognized and degraded by SCF, and then at this point in the cell cycle, APC, which is your other ubiquitin ligase, that's big in cell cycle, that's inhibited, it's shut off because it's CDC20 coactivator is not around anymore. It's inhibited. So APC is off and geminin levels rise because geminin is a substrate of APC. Geminin levels rise and bind to and inhibit CDT1. So that's how you have those things inhibited, and that's what prevents formation of the pre-RC again. You prevent formation of the pre-RC, you don't get constant initiation of DNA replication. Okay? You can see how if you didn't inhibit these and you allowed this to constantly form on origins, what's to stop constant activation of DNA replication from origins that are already activated, right? If your S cyclin is there and your SCDK is active and you have this constantly forming, can you imagine what your genome would start to look like if you kept on getting things recruited to your origins and you kept on initiating DNA replication? You'd have this craziness going on, right? That makes sense? So you really want that. That's why it's important to regulate DNA replication only once per cell cycle. Okay, any questions about this? Okay, so initiation. So we've really talked a lot about initiation, but it's pretty important. It's followed by priming and synthesis. And this is a picture I've shown you a few times. It's a picture that you're looking at as you go through this week's recitation. And the thing I'm really trying to drive home, and I felt that even though I mentioned this a few times, a lot of people did not get it because on Monday's recitation, a lot of people were drawing replication forks incorrectly, and I was really thankful they were doing it incorrectly because it allowed me to point out what they didn't understand, and there were a lot of people who were like, oh, and I think that's always a good thing. So what you need to notice is you have your helicase interacting. It's all kind of interacting. Your helicase, your DNA polymerase, your clamp, your primase, your sliding clamp, clamp litter. It's all in one complex and working together and moving in the same direction. This is counter to what you guys learned in 201 and 309, where people just showed you DNA polymerases going in different directions. And I just want you to realize this is correct, the other one is not. And I talked to some people who teach 201 and 309, and the consensus is that it's such an introductory level class that it's overwhelming to put this level of detail in at that early phase. So that's why you get it from me. So leading and lagging strand synthesis is coupled. It's happening at the same place at the replication fork. Okay, so I've got a few, yes?
Going back to that, is RNA primase associated with primase?
Yes, your primase is right there. A little blue thing, and that's your little primer. So here's your primers. So it makes a primer, and then this is all single-strand binding protein coated. This is our RNA primer. And so eventually what happens, this thing is moving forward, and your primer ends up interacting your polymerase, and it copies in between your primers. And there's your lagging strand synthesis. Make sense?
I'm just confused. The DNA primase was set RNA primers.
It's an RNA primase.
Oh.
Yeah. So your primers are RNA. Yeah. Surprising. Weird. I wonder why. Your DNA polymerase is polymerizing your DNA, and later your RNA primers get removed and filled in by another DNA polymerase and
I'm
lyse.
just confused because it says DNA primase.
DNA primers. Well, it's DNA primase is priming. It's because it's priming on the DNF, but it's making an RNA primer.
Okay.
Yeah. Yeah. DNA primase makes an RNA primer. Okay. Good. Anything else about this? Questions? You guys will really get it as you draw it in recitation. Okay, so I've got some videos. I was looking for some and I kept finding bad ones, and then I realized I had found some last year. So hopefully audio works. Let's pray. Everyone say a quick prayer. whoever you believe in, if you just believe in the spaghetti monster or whatever, say a prayer. This is a really good one. It's all going to be available to you on your on Blackboard, so you can watch it.
The process of bacterial DNA
course not.
replication involves a number of proteins coming together.
I'm going to have to narrate for you.
DNA replication begins at a single defined DNA sequence of 245 base pairs called OEC. A protein called DNA-A increases in concentration.
Okay, so I'll just narrate.
And gets ready for cell division.
So you've got, this is in bacteria, right? So you've got your DNA-A that binds to the origin. Remember there were those two regions. There was the DNA-A binding region and the AT-rich region. It binds and actually distorts the DNA, and that helps to open it.
The opening in the DNA allows protein complexes to enter the replication bubble and bind to the single-stranded DNA.
So that's your helicase, your DNA-B, and DNA-C. Helicase and helicase loader are going to be loaded onto your DNA.
Also called DNA-C. The DNA helicase loaders open the DNA helicase protein rings and place the rings around the single-stranded DNA.
Notice they're rings that are placed around the single-stranded DNA. So people were drawing helicase as something that was like shooting through the DNA, but it's actually wrapped around, I believe, your lagging strand. So then primase is recruited by helicase. Makes your RNA primer. That is the source of your 3'OH.
The main replication polymerase in E. coli is called DNA polymerase III.
So notice how this whole complex is recruited together, right? You've got two DNA polymerases
Complexes called clamp loaders.
with the clamp loader and the clamp.
Other protein complexes called sliding clamps.
And each of those is going to go to one replication fork in the replication bubble. See that?
It then places an attached DNA polymerase III complex next to the sliding clamp. The sliding clamp holds the DNA polymerase.
So here's your sliding clamp for your leading strand.
As the polymerase synthesizes new DNA, nucleotides with complementary bases to the template strand are added one by one in the 5' to 3' direction. The synthesis of DNA in the direction of the fork occurs continuously to the end.
So see how it's going out this way? See how the whole complex is together and you have this looping out going on in your lagging strand to create your Okazaki fragments. Notice the whole complex stays together. DNA polymerase is not moving away from each other ever.
In the leading and lagging strands. Note that the template strands are anti-parallel with their 3' and 5' ends oriented in opposite directions. Because DNA polymerase can add nucleotides only in the 5' to 3' direction, the leading strand grows continuously in the direction of the replication fork, but the lagging strand.
So you have the continuous synthesis on your leading strand and the discontinuous synthesis on your lagging strand on both sides. Remember, you've got two replication forks. The same thing is happening on both sides.
Binding proteins, which quickly co-expose single-stranded regions of DNA.
So you've got the single-stranded binding proteins that coat the single-stranded regions of DNA.
Although single-stranded DNA binding proteins are present throughout replication, for simplicity, we will omit them in the rest of the process. DNA replication continues as the DNA polymerase on the lagging strand meets the 5'.
So notice the lagging strand.
Causing the polymerase and the sliding.
Once it hits the next primer, it releases.
After DNA helicase has moved approximately 1,000 bases, a second RNA primer is.
And so helicase is moving forward, and then primase is recruited and makes another primer all the way down here. And then you have recruitment of the polymerase again, and the Okazaki fragment is synthesized. And so that's your lagging strand can be fairly complicated. So this I felt showed it really nicely. And so it just keeps going and going.
Note that the lagging strand now consists of Okazaki fragments with a segment of RNA at one end.
So you've got this RNA primer stuck in here, and it needs to be removed.
RNase-H.
So RNase H removes it.
Another enzyme called DNA polymerase 1
A different
uses the
DNA
3'
polymerase fills in that
molecule
gap.
of the adjacent Okazaki fragment to fill in the large gap with DNA nucleotides. Finally, an enzyme
And then
called
DNA ligase ligates
closes the
the
remaining
two
ends of
together.
DNA, leaving a continuous DNA molecule. In this way, an E. coli chromosome is replicated at 2 replication forks, all the way around the circular molecule.
There you go. Okay, so that was one. I have another movie for you that I'll get to narrate. Oh, actually, so this I just put up because on NCBI I keep pushing people to not limit themselves to Alberts. And so I put this up because this is all explained very nicely in molecular biology, molecular cell biology, which is by Lotus. And it's free, it's online. So when you read something and it doesn't speak to you and you just can't understand it because of how it's written or whatever, don't limit yourself to one textbook. Look at everything. And so you have the link for this. You get it anyways. When you go to NCBI Books and put in anything, it'll bring up all the books that have information. And sometimes you might need to read about it by a few different authors to truly get it. So I just put that in so you have it. And so here is that it? Here's another one. This is a YouTube video, and this is put out by Garland Science, who's the people who publish our text.
Two DNA polymerases collaborate to copy the leading strand.
And so it's just another way to look at the same thing, so you guys can just...
In this picture,
The
the
way
DNA
the loop
polymerase
is,
that
and
produces
there's a
the
different
lagging
orientation,
strand
but it's
has
the
just
same-
finished
the
an
same
Okazaki
purpose.
fragment.
That's your
The clamp
parental.
that keeps the lower DNA
This is
polymerase
exactly the
attached to
picture
the
you
lagging
have in your
strand
book, right?
dissociates, and the DNA
'Cause it looks
polymerase
like ours.
temporarily releases the lagging strand template DNA.
But see, there's your
As
primase.
the
It makes
DNA
your primer.
helicase continues to unwind the parental DNA, the primase becomes activated and
Helicase looks funny
synthesizes a
here,
short
doesn't it?
RNA primer on the growing lagging strand.
So right, your primase is making your primer, and the thing is moving
The DNA
around
polymerase
until it
binds to the
gets
DNA
to
again and becomes locked in by the clamp.
the polymerase,
The
and then you get
polymerase uses
synthesis
the
on your
RNA
lagging
primer
strand.
to begin a short
Make it
copy of the
Okazaki
lagging
fragment.
strand template DNA. The polymerase stalls when it reaches the RNA primer of the preceding Okazaki fragment.
I
and the
believe
entire
this is
cycle
really inaccurate.
repeats.
It is a ring that's around your DNA. I don't know why they chose that one. Maybe just for simplicity. This one has really nice music in the background. Right, so the single strand, I got a lot of questions about the single strand binding proteins. Notice they're coming off as polymerase, as the new nucleotide. They're really just there to protect that single strand of DNA from nucleases. Probably also to prevent activation of the DNA damage response, I guess. Right, so notice the clamp. Notice the clamp is not going anywhere in the leading strand, right? But it keeps on coming and going on the lagging strand, right? Because you have continuous synthesis on your leading strand and that discontinuous synthesis on your lagging strand. Notice that the primase is only working on your lagging strand. You only need it once on your leading strand, right? So you guys have that. You can watch it over and over again to your heart's content. I have to figure out the audio in this classroom. Okay, so there's polymerization. So we understand initiation, we understand activation, right? We understand polymerization of leading and lagging strand. And so now we need to terminate. And so it's really simple in bacteria because you start in one place and then you end in one place, right? So the replication forks are going to meet at the opposite end from where they start. Makes sense, right? Assuming they both move at the same rate. This happens in bacteria and viruses, right? Because viruses have to replicate their genomes too. specifically with circular DNA. Termination results in linked chromosomes, so they'll be linked kind of like chain link, and so that's called catenation or catenated, and they need to be decatenated, and that's done by topoisomerase. Remember we learned about strand passage, and so that's how that happens. The type 2 topoisomerase. Good, we're doing good on time. Okay. So in eukaryotic cells, it's a little different, right? Because can you just have termination opposite from where it begins? No, right? Because you have a lot of origins. So basically, all that happens is the origins fire and they run into each other until all of the DNA is copied. But you do end up with kind of a big mess. And that results in all these daughter molecules being intertwined, and they're resolved also by topoisomerase. Right? So you get replication, see there's origin, and you've got just ends up with things being intertwined based on different origins firing at different places at different times. And then topoisomerase helps them entangle. Okay? Any questions? Okay, so let's go back to the end replication problem, right? So we talked about telomeres in terms of structures of chromosomes. Let's talk about it a little bit more, right? So we talked about DNA replication and the leading and the lagging strand, right? And so do you notice your leading strand is no problem because you've got your primer and you get continuous synthesis until you're done. But your lagging strand is the real problem because you've got this RNA primer there that needs to be removed, and then there's no way to extend it, right? So, and if we don't fix this problem, then we'll get shorter and shorter chromosomes over time. Is this a problem in bacteria? No. Okay. Why is it not a problem in bacteria? I'm going to call on someone. I haven't called on anyone today. I've been lazy. Brianna. Where are you? Oh my gosh. Where is she? That's okay. You're her keeper. Okay. Kelly. Why is the end replication problem not a problem in bacteria? You can phone a friend. Because it's circular, right? Right, just like what I said, you continue until you're at the end. That's no problem. Really, the problem here is the primers serving as a 3' OH upon which to add your nucleotides, right? And so once you remove this primer, you've got no 3' OH to add your new nucleotides to. So that's why it ends up shorter there. And so that's what you're seeing here. So you have your parental, so well, your basically whatever was your leading strand is going to be synthesized continuously and be full length, and whatever your lagging strand is going to be a bit shorter. And so this is another way to look at it and how it's happening, right? So this, I don't like, this is from the old textbook I used to use. It shows the end replication problem nicely, but I don't like how it shows the whole replication complex. It's associated, but it shows it with a lot of space in between, which bothers me. But notice as you get to the end, on your leading strand, it's no big deal, right? Because you're just gonna have replication to the end. On your lagging strand, you're gonna end up with a primer. So here's a primer, and you get replication, right? You're gonna end up with a primer here that's removed. So that's your end replication problem. And so that's solved by telomerase. So telomerase, we talked about already. It's an enzyme that synthesizes DNA using an RNA template. Where's that RNA coming from? Dennis, I just saw you. Ah, right in front. So where's the RNA coming from? The RNA template. You can phone a friend.
And telomerase.
It's the telomerase, right? So it's part of telomerase. It's actually part of the enzyme. The enzyme telomerase is made of RNA and protein, so it carries around its own RNA template. And so the way it works is it recognizes. So here's the way your end of your chromosome looks, right? You've got your parental strand, and then you've got your new, and this was your lagging strand, right? And you have the end replication problem right here. And so telomerase, here's your telomerase enzyme with its own RNA template, and it's going to actually recognize the end. There are these repeats at the end, so it's going to recognize the end of your parental strand and extend it. It's extending your parental strand, right? So it makes it longer. And then DNA polymerase comes in, a different DNA polymerase comes in and uses that as a template to lengthen that lagging strand that was too short. You still always end up with inequality, right? Your parental strand, or what your leading strand was, is always going to be a little bit longer because it was extended by telomerase. And what it actually does is it loops around and binds to the other strand of DNA. And I don't know if I put a picture in. No, I didn't. I'm going to put a picture in for next time. Yes?
Why does telomerase only bind to those two bases and not the full complementary sequence?
Because what's happening here is it's acting as a template to fill this in. So if it binds to the whole thing, then there's no template available. So the question was, why is it only binding to two bases and not the whole sequence? And that's because it's acting as a template. So it's the template and it's adding on additional nucleotides. So this is going to be a G, 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 T, T, G, like that. And that's why you have these repeats at the end. They're based on the template that is part of telomerase. Okay, any questions about this? Yes?
So is there another primer put on the end for DNA polymerase to start from?
Right, so that's always been my question. Like, wait a minute, isn't the end replication problem still a problem? And telomerase isn't actually resolving much of anything? And I think the way it's explained, it seems that way. But, and I don't think it's completely understood, but it's believed that this DNA polymerase brings its own primase. And so it's not the DNA primase involved in replication. It actually brings in a primase that provides that 3' OH. That's what it's believed, because it doesn't work otherwise, right? Right. I was looking through the textbook and I was like, I'm so unsatisfied with the explanation. And it said it's believed that, so it makes me think that it's not known. But maybe, we haven't done an extra credit in a while. So let's turn this into an extra credit, right? So I'm telling you that it's believed that this DNA polymerase that comes in and fills in the end of a chromosome brings a primase. But what does it bring? So the question was, wait a minute, there's still no 3' OH. How is this DNA polymerase adding these nucleotides? There's no primer, right? Well, I don't know. There must be, otherwise it makes no sense. So you guys need to tell me, what is the mechanism of addition of the telomere ends, not by telomerase, but by the DNA polymerase that comes and uses the telomere DNA as a template? Questions about the question? Questions about the question? Does everyone see where the confusion was? Yes?
Might not be saying it right, but then also when telomerase, the DNA polymerase is bringing its own primer to add.
So the question is, wait a minute, what's the point of telomerase if the DNA, if the DNA polymerase, a DNA polymerase, is bringing in its own primer? Well, if you think about it, if there's no telomerase to extend the ends, through repeated replication, it's going to get shorter and shorter. And so, yeah, it might fill in a little bit, but telomerase takes many repeats, so
it starts
significantly
on your
extends the template. It basically creates a very long template that is filled in.
So right there, that's not showing the effect effectively just like the two inside the atomic. Like where they're showing it starting 2 bases after the telomerase at the sequence.
So it starts
Probably
here.
a lot.
It starts here and then it keeps going. Maybe it goes down to there.
I mean, where DNA polymerase is starting, it's actually much further than just 2 bases inside where telomerase is at.
Probably. Right. So the question was so is this accurate that it's just DNA polymerase binds here and extends? No, it probably binds like it has some kind of primase or something that puts something to start it, probably way at the end. But you do not start right at the end. It's not 2 equal lengths. Always you're going to have this overhang, and it loops around and ends up binding through like a strand invasion kind of mechanism to the chromosome. Yes.
That's what I was going to ask. I was wondering what happens to the extra end. It just binds the.
So this part, right?
Yeah.
It forms a T. It's called a T loop. I thought I put a slide in. I'm going to put it in. I'll start with that next time. And it actually loops around and invades in here, and that's how it forms the end of the chromosome. Yeah, because you would think keeping this just hanging here would make it highly susceptible to nucleases, so it loops around, and you don't have that single strand DNA. Any questions? Good questions today. Okay. Well, I think I've got maybe one more slide. So this is just comparing how different organisms overcome the end replication problem, right? So bacterial, so if you think of different organisms, they have different types of genomes. Bacteria have circular chromosomes, so that's how they overcome their end replication problem. Then you have recombination in mitochondrial genomes in some bacteria. Drosophila telomeres, they have transposition, so this is definitely unique. Basically, different organisms have overcome this problem in different ways. Different viruses will sometimes they'll form hairpins at the end, and then we add these telomere repeats. So it's eukaryotic mechanism for overcoming the problem. Okay, I think that's it for today. Any questions? Nope. So I guess I'll see you guys on Friday or in a few minutes.